Swayam Prabha. Digital India. Educated India.
Next lecturer, Dr. Hemachandra Kottamarthi. He has done his BSc in Maths, Physics, Chemistry from AV College of Arts, Science and Commerce, and done his MSc in Chemistry from University of Hyderabad. He has completed his PhD in Biophysical Chemistry from Tata Institute of Fundamental Research, Mumbai. His research interest lies on biophysical chemistry, mechanobiochemistry, protein folding and unfolding, etc. On behalf of everyone, I welcome you, sir. I am sure that he will enlighten us with his knowledge. So please give a round of applause to him.
So before I tell you something that you are probably like a bit aware of it and you want to know more about it, so I want to, as somebody has mentioned here, so I'm in the Department of Chemistry here, but I work in the interdisciplinary areas. In the sense, I really don't work in the traditional chemistry that you are aware of. I work in the area of biophysical chemistry, where I use my knowledge on chemistry to understand biomolecules and to look at how they could be actually enhance our understanding of life in general. Okay, so some of you have taken biology in your +1 and +2. Some of you have not taken, but it doesn't matter, because what I'm going to talk about today is a very simple biology, and which you will be everybody would have kind of gone through this kind of biology at some point in their life. Okay, so today I'm going to talk to you about a system called CRISPR. Okay, so I'll once the slides come out, I'll tell you. Okay, this is called a CRISPR, and this is actually a tool. But what I want you to focus on is not just the research, just look at how the ideas came about. In a sense, this is one of the technologies that has been developed. But look at how the idea for that technology came about. Like, this is how the research goes in. You don't actually wake up suddenly one day and say that, okay, I have invented something, right? You actually go through a certain technology. For you to get into some technology, there is a lot of science involved in that. And something that is completely unrelated. When you are doing some science, you would not have thought that this will be applicable for such systems. So you'll see how an experiment on trying to understand bacterial defense mechanisms has led to genome editing. Okay, so we will talk about that, but you will see how they are completely different realms of science. One is a very fundamental, you're trying to understand how bacteria bacteria also have infections. Although we are getting infections from bacteria also get infections from virus, right? So how does a mechanism that was developed by bacteria to evade viral infections actually has resulted in a completely different technology which the humans are making use of? And the other thing that I want to emphasize here is that, so as again I said, so at some point you need to understand all the basic sciences in detail, but never have a boundaries in the sciences, in the sense like this is physics, this is biology, this is chemistry, this is maths. At some point you realize that what you want is to solve a problem. It doesn't matter whether you are a physicist, chemist, mathematician or a biologist. Biology has some of the best problems to solve. Physics and chemistry has some of the best tools to solve. So biology is full of problems, and all the tools that you apply are from physics and chemistry. So as you go into higher levels, so you don't have a barrier that this is physics, this is chemistry, this is something, okay, because it's all about solving a specific problem and how do you get ideas from various disciplines. Okay, so that is what this story is about. But there is a story involved in that, but of course there is a science involved in this. So we will see how it goes. Okay, so I have shown you something. Okay, I have shown you some 4 different pictures here to start with, right? One is eradication of some infectious diseases. The other is increased food production. Then you have organ resources and rebuilt extinct animals. Now you think, can something correlate all these 4? Is there any technology or is there something that can be used to correlate? These are very very, very different kind of problems. One is trying to understand or eradicating the infectious insects, and one is to rebuild extinct animals. There are very different realms of science or realms of problems that we are trying to address, right? So, you think there is something common technology or some common thing that can be used to do all of this and much beyond this? Recombinant DNA technology. Good. You're close by, but it needs slightly advanced of it. Yes, you're sure. You're right that there is something called recombinant DNA technology, which we'll be talking about. So all of this, any living system has something in common. What is that? DNA, right? Now, if you can edit the DNA, right? If you can edit the DNA, right? DNA is also like I'll talk about it, but if you can make changes in the DNA, if you can edit the DNA, any properties that you're seeing in a physical systems can be done. Like you can, now a particular production of a food production can be increased by changing some of the genes in a plant, right? Or if you want to get rid of the infectious diseases, you can modify a gene so that it doesn't bind to a parasite or it doesn't have a reproduction, right? So anything that you want to do with the life can be modified by something called as by editing the DNA. So the story that I'm going to talk about is on genome editing. Again, so this is a genome editing topic which is coming from a biophysical chemist. So my perspective of this will be very different than a traditional biologist. Okay. Now, what is genome editing? So as I said, how many of you know what are genomes? I see 3-4 hands. Okay. Then okay, let me go back a word before. How many of you know what is DNA? Okay, at least this is something that everybody knows, right? So genome is nothing but a more complicated version of DNA. Okay, now if I have to basically, so I'm going to talk about a story which is called molecular scissors. Okay, so how this, to edit something, if you have a paper or if you want to modify something, what will you do? You use scissors and cut into the shape that you want, right? So we are going to talk about a topic called molecular scissors and how these scissors are being used to edit DNA. Okay, but before going into that, because I know a lot of you are not probably biology students, so I want to go into basics of what I am talking and then go into the advanced topic. Okay, so if you look at genome, let's start with, before I say what is genome editing, you should know what is genome, right? Like to edit something, you should understand what it is, right? We all know that whatever the physical traits that we have, my color of my eyes is black or somebody's color is blue, right? Somebody has a black hair. In India, everybody has a black hair, but if you go to abroad, if you go to foreign countries, they have a brunette, they have a golden color hair, right? You have different varieties of color. So any physical trait that you are seeing is basically, where is coming from? It is coming from a genome. There is a code that is there inside your body that will tell you how you look like how you should look like, right? Like you should have probably like some people have 6 fingers, some people have 5 fingers, right? How do you color? So all this information is there when you are, I would say if you want to put it in terms of programming language, you have been programmed to be this way. You will only be this way, right? You cannot be any other way because there is already a program inside you, but only to some extent. Okay, you can always change the parts of it. But if you want to understand this, so basically any physical trait, not just trait, even the diseases that you get are basically from your genome. For example, people from the southern part of your Vindhyas, whom we typically call South Indians, very prone to diabetes. Our genome, our genes have been made in such a way that we are very prone to diabetes, right? There are some other part, for example, who are very prone to other kind of diseases. So even the kind of diseases that you inherit are also determined by your genome. So a part of your life is already programmed. You only have to live the other part because you cannot change the part that has been programmed. But this is a topic that I am going to tell you how you can even change the part of the life which has already been programmed. Okay, so if we talk about that, so genome is something, a code. If you're all computer science students or if you're a biology student, it's basically a code that will tell you what is your height, what is your color, what is your weight, or not weight is not something that is not dependent on that. So what is your, basically what diseases are you prone to, this kind of basic information. Now if you want to edit any of the living organisms, if you can edit the genome, you can edit yourself, right? But that is little too far-fetched. You can't edit yourself, but I'll tell you some of the techniques that are used to edit DNA. Okay. Now, if you want to compare DNA organization, before we go into genome, now just imagine that DNA is like a storybook, or your life, the entire genome is the story of your life, right? If the entire genome is the story of your life, this is the organization. There are chromosomes, which are like your paragraphs. Then there are genes, which are like your sentences, and then there are DNA, which are letters. So basically DNA is the letter, right? And you make a lot of letter, you will make a sentence, right? That sentence is called as gene. That has a meaningful, so a DNA doesn't have any meaning on its own. It's just like letters. You put in letters, it is letters. It doesn't have any meaning. Now you write letters in a systematic way, you get a sentence. Sentence has a meaning, right? So a gene is like your sentence. Now every gene can produce a specific protein. So when a gene will give rise to a protein, right? So it is like basically you have DNA, which are alphabets, you are making, or letters, and then you are making these letters in such a way that they have a meaningful, they form a meaningful sentence. That is called as gene. And if you make a lot of these sentences, what will form? A paragraph, a short paragraph or a long paragraph, which will have a continuity and which will make a sustainability on its own. That is called as chromosome. And you all know that half your chromosomes come from your mother, half comes from your father, right? So you have as a human being, so you get like part of their qualities, part of mother's, part of father. So a chromosome is like a it's like a paragraph, and then you have all the paragraph mixed. That is called as genome. So if you want to have an analogy, genome is your storybook and the letters are DNA, right? And then you have like whether you want to have a sentences that are called genes, which basically make a meaning in that, and then a paragraph which makes a complete meaning in a sentences by themselves do not tell you what is coming after or what is coming before. Whereas a paragraph is generally a self-sustained, in the sense if you read a paragraph, you can understand a bit more about the story, right? So it is a simple analogy of genome would be something like this. A genome is the story of your life and DNA is the alphabets. Now if I want to edit genome, what should I edit? I should edit my DNA. So you edit the alphabets, then you basically if you want to edit anything, the basic unit that you have to edit is your DNA, right? So today we are going to talk about how we edit DNA and how editing DNA is going to have an effect on the overall genome. So just to, yes, somebody had a question? Okay, so just to give you a rough idea, right? So this is basically a rough estimation. A human being has around 3 billion base pairs. Now how many of you know what are base pairs? So you know what are base pairs. If not, that also I'll tell you. So a DNA has something called as, there are 3 components in DNA. One is base, one is sugar, the other is phosphate. Okay, so we will talk about that because but just for you to understand, so basically there are 3 billion base pairs, DNA base pairs in a human being, and that results in 21,000 genes, and these genes will become 46 chromosomes, and these 46 chromosomes will become one genome. Okay, so your story is in this one genome, which is basically from the 46 chromosomes, 23 from your mother, 23 from your father, and then there are each of those basically there are a total of 21,000 genes. Each gene would code to certain protein. That means it is a sentence on its own, and the starting material or the alphabets for that is your DNA, which is your 3 billion. Now you can see that is for humans, but if you see the other organisms, I have written like few other organisms, even a simple bacteria has around 4 billion bases. So it's not like a 10 bases or 20 bases. Even a simplest living organisms, for it to self-sustain, like you have to have a process from where you take birth to the end of that, right, life. Even for a bacteria, it has to divide, and after division, it will again further divide, and in between there are a lot of other processes. So this, all the information is coded in this. So we will talk about how do we edit this. If you understand that by editing the DNA, you can edit the genome, then we will move to the next, basically what exactly is your base pairs, right? Now, so today, depending on how it goes, so don't worry about the structure of today's talk, but let us look at the basic thing, right? How many of you are aware of this picture? Some of you are aware, right? So basically, this is called a central dogma of molecular biology. Now we all say that, okay, so DNA is a basic essential thing, but you don't have everything in the DNA. Now DNA doesn't function, right? You need something that is functional. So DNA actually undergoes what is called transcription to give what is called as RNA, and RNA will translate to form proteins. Now proteins are called as workhorses of the cell. Workhorses in the sense, anything that you do, I'm speaking, I'm listening, my hands are moving, anything that is happening in my body, what is the unit that is doing? That is proteins, right? So for example, how many of you are aware of proteins? I guess everybody should be here. In what context are you aware of proteins? In the cell, how it forms from the amino acids. And what other contexts are you aware of the proteins? Nutrients, carbs. So most of you are probably aware that proteins means it is some kind of nutrients, just like your carbs, there is proteins and you have it as your diet, as a part of your diet, right? That is what you think. But no, there is much more beyond that. Like you take proteins to increase your amino acid compositions, increase certain amino acids. But do you know that all the functions that we are doing, like for example, I am hearing, right? There are proteins that are communicating the sound waves that I'm getting into my brain, right? I am seeing there are proteins, there are crystalline proteins that convert the optical image into or the optical signal into an image, right? So all these are being done. So for example, if you look at proteins like chitin, which is a muscle protein, right? It is it's one of the longest proteins. So it will help you to have a structural integrity. So proteins are not just nutrients. Proteins are well beyond that, in the sense what you are is proteins, right? All the functions that you are doing as a human or as a life is being done by proteins. So proteins are called as workhorses of the cell. That means they are the ones that perform a lot of activities, or I would say all the activities that you do in the cell are performed by proteins. So, but proteins, when you are born, you are not like basically, so when information is carried out, or when information is transferred from one organism to other organism, the information is not transferred in the form of proteins. The information is transferred in the form of DNA, right? Then that DNA converts, or then that DNA becomes RNA, which is called as transcription, and that gives a functional things which is called as proteins. I'll give you an example, right? All of you have heard of COVID-19. I'm sure nobody is there who did not hear of COVID-19, right? What is the virus that caused it? SARS-CoV-2. Okay, and some of you have heard that what kind of virus is that? So basically, the viral particles do not have a life on its own. How do they survive? They inject their genetic material, right? So basically, that means they don't have a machinery that can produce proteins or these things. They have a bit of machinery, but they cannot survive on their own. So what do? They just inject the genetic material, either a DNA or RNA. Okay, so they inject the genetic material into the host cell. For example, I am the host cell. They inject into it. My machinery thinks that this is the DNA, and that converts into RNA if it is a DNA, and then that converts into proteins. Now the proteins that are produced are not my proteins. The proteins are viral proteins. So that takes over the cell. Our cell do not know what to do with that. So those proteins take over the cell, and then ultimately you get all kinds of infections in the sense something that is not relevant to your body, right? So the information is transferred from one organism to other organism. In most of the cases, it is transferred as DNA. And then if you have a DNA, then you get proteins, and proteins are the functional forms of this. So you probably would have seen this. This is how an RNA looks like. DNA has a double helix structure. Okay, RNA is a single stranded one. Okay, do you know the chemical composition of DNA and RNA? What is the main difference between DNA and RNA? So let me tell the full form. DNA is deoxyribose nucleic acid. RNA is ribose nucleic acid. Right? What is the difference? Okay, so there are two OH groups in RNA. There is only one OH group. If you want to look at the structure, I said DNA is composed of three parts, right? One is your base, which is called either purine or pyrimidine kind of bases, and then there is sugar, which is constant. These are called ribose sugars. You know sugars, right? Which are carbohydrates, glucose, fructose. Similarly, there is a ribose, which is a 5-membered ring. So ribose sugar, and then all these units are joined by a phosphodiester bond. Now DNA is nothing but a chemical moiety which is made up of 3 units. One is called base, which is either an organic molecule, something like this, which is a pyrimidine or a purine kind of system that is attached to a ribose sugar, and then that is attached to a phosphodiester bond, and this forms a long chain. How long chain? As I said, there are DNAs which can go to few million base pairs. Okay. So it is very common to find base pairs in like a few thousands of base pairs. Very, very common. Any bacteria you take, it will have like few 20,000, 30,000 base pairs. That means they're all linked by this phosphodiester bond. So basically there is a sugar. So all these things, if you see, they all differ by only bases. So the sugar part and the phosphodiester bond part is constant for everything. So only the sugar part changes. Only the base part changes. In one case it is called adenosine, guanine, cytosine and thymine. So these are called AGCT, or you can also call ATGC. Basically the DNAs, as I said, there are only 4 alphabets. We have 26, right, letters in an alphabet. In English alphabet has 26 letters. But human living system alphabet, right, has only 4 letters. So our entire code is written only in the form of these 4 letters, A, G, C, T. And A is basically a DNA with adenosine base pairs. Then you have, these things are again common, the sugar and the phosphate, guanine, cytosine and thymine. Now this is your DNA. And what is RNA? RNA is very similar to that. So for example, this is DNA. DNA, if you they form what are called as hydrogen bonding networks. How many of you know what are hydrogen bonds? All of you know hydrogen bonds. So what is a hydrogen bond? Hydrogen and a highly electronegative atom, right? So this DNA, the double helix of this DNA is stabilized by simple hydrogen bonds. So they are probably one of the weakest bonds, but still because there are so many of them that it stabilizes the entire structure of that. Right? So basically you would have the entire DNA that is basically bonded. A will bind to T, G will bind to C. This forms a DNA. Now the difference between DNA and RNA is very, very small. So you see this. Okay, let me see. So you see this OH for the sugar. In DNA there is only one OH. In RNA there will be 2 OHs. So DNA is called deoxy. So you will lose one oxy and then you have another OH in RNA. Now this entire thing will cause it to make, so this single OH will make it to form a double stranded DNA. But if you have 2 OH, it will make it form a single stranded DNA. Why do you think so? No, it can form a bond here, it can form a bond here, right? So for example, the hydrogen bond here is only between the bases, right? Your sugar part is not actually involved in the hydrogen bond. So if you see this, your bases are the ones that are forming the hydrogen bonds. Whereas sugar has a OH, it doesn't form a hydrogen bond, right? It is not involved in the hydrogen bond. But still, your DNA is a double helix. That means it will allow it to form hydrogen bond. But RNA is a single helix. You cannot form a hydrogen bond. Or it doesn't form a double, 2 of the DNA pairs do not come together. Have you heard of a word called steric repulsion? What is steric repulsion? See, I am trying to correlate the biology that we are learning into the actual chemistry that is happening. What is steric repulsion? If there are 2 very bulky atoms or bulky molecules, the electron cloud around the electron, in this also there are a lot of electrons which are negatively charged, in this also there are a lot of electrons which are negatively charged. So negative charges repel each other, so that will make this bond kind of repel each other. So if there is one more OH here, assume there is one more OH here, and basically there is one more strand like this. So OH, the OH will form, these are 2 bulky molecules, they will start repelling each other and your double helix is no longer a stable one. So that is why you see how nature has developed. It has removed the OH so that it can have a double helix. If you have the additional OH, that is called RNA. So to store something, again if I go back to this, now if you look at a chemical reaction, you have a starting material, you have a product, you have an intermediate, right? This is a general chemical reaction. Your starting material is DNA. So in the life form, everything is stored as DNA because it is a very, very stable structure, because two of the segments have come together and they are binding. It is very difficult to separate them out. So it is going to be a very, very stable structure. So everything in the life is stored in the form of a DNA, which is a stable structure. Now if you want to react something, it will be RNA. RNA is a highly reactive molecule because it is in a single stranded form. It can easily form any hydrogen bond or other kind of bonds with that, right? So storage is always in the DNA and your final product is protein, which is the functional form. So basically now you have a DNA, which you have an RNA. DNA always forms hydrogen bonds between two strands, which is allowing it to form a double helix, stable structure, and RNA doesn't form that, and this is how RNA looks like. You have an additional OH group. So this is to re-emphasize all the whatever we are going to study. I'll be moving into the bio part, but although I'm saying bio, what we are dealing with are nothing but chemical molecules. All these molecules are chemical molecules, but we sometimes write them in a simple way so that we don't complicate. We don't want to write these complex structures every time we draw, right? So we will write. If you want to look at a DNA, nobody is going to, or you generally don't see people writing all the bases and then the sugars and then the phosphate. So nobody is going to do that, right? So you just write it as like some kind of double helix, right? And then you have basically, you are saying that this is one DNA strand or this is other DNA strand, and then the hydrogen bond. Simply we are representing it, but none of them are threads. They are actually a huge chemical composition that goes into that, right? So in order to edit, again going back to the original one, why did I start this? I want to tell you about genome editing, and I said to edit the genome, we have to edit the DNA. And what is it that we have to edit? We have to edit some of these chemical moieties that are there. That will result in DNA editing, and we are mostly going to edit what are called as these phosphodiester bonds. Okay, so from now on, okay, I am not going to draw these complex structures. I'll just represent DNA as this railway track. Okay, DNA I am representing as a railway track, just like so showing that this is one DNA strand, this is other strand, and they are bound by hydrogen bonds. Okay, that is what I want to represent from now. And then if I have to represent the RNA, I'll just represent by a single strand, and these are again hydrogen bonds. So basically it is ready to form the hydrogen bonds. It didn't form, but in principle it can form. So I'm going to use this notation in any of my future slides to represent DNA or RNA. Okay. Now if I have to, I said I have to edit my DNA, right? That is what the topic is today. Now if I have to edit the DNA, the bond that I have to make or break, when I have to edit, the first thing that I have to do in editing is cutting, right? And then you paste something new or you change it. So for me to cut, if you see this, all the DNA base pairs are joined by this phosphodiester bond. Can you see this phosphodiester bond here? A P double bond O. That is your phosphodiester bond. Now if I have to remove all these are only, none of the bases are actually interacting here. None of the sugars are interacting. All the interaction is through phosphodiester bond. Now if I have to edit my DNA, or if I have to remove certain bases or to add certain bases, the thing I have to remove or add, basically my reaction has to be on the phosphodiester bond. So this is the reaction that I am going to do. So this is your DNA with a sugar and a base and a phosphodiester. Now if I use something that hydrolyzes this phosphodiester bond, that will break into two different components, right? That is called as editing. So basically what I'm doing is a chemical reaction where the phosphodiester bond is being cleaved or a phosphodiester bond is broken. Now this phosphodiester bond can be, there are two, okay. Before that, so if you have to do a chemical reaction in the body, what kind of molecules perform chemical reactions in a body? Enzymes, hormones, okay, reactive enzymes, some reactive molecules, charged molecules, okay. So, but any chemical reaction in a body, I'm not talking about a human body, in any living system, is done by what are called as enzymes, okay. Have you heard of the term enzymes? All of you have heard of enzymes. right? What are enzymes?
Biocatalysts.
Okay, that's easy, right? Enzymes are biocatalysts. What is so interesting about that?
The speed of the reaction.
Okay, the name itself indicates catalyst, right? So it increases the speed of a reaction many fold, right? So we will see how many times. Okay, and what else? Is there anything else that is there?
It does not involve itself.
Okay, because it is a catalyst, it involves in the reaction, but it doesn't consume. Basically, you get the reaction back or enzyme back, but it has to involve in the reaction, but it is not used up in the reaction. Okay, and what else?
Reaction takes place in the active sites in the protein foldings where the enzyme acts.
Reaction
Take place in the protein foldings active sites where.
So basically, they actually perform this reaction in a specific site called active site. Okay. Okay. Now that is a point I was looking for. So basically, enzymes are there always in our body. Now, if you do a chemical reaction in the body, like for example, if you want to do a hydrolysis reaction, acid hydrolysis or something, you add an acid. Now, whatever are the reactants, all of them will be reactive to that, right? Now, what enzymes do is, they kind of look into so if you want to only break the phosphodiester bond, DNA also has a phosphodiester bond, RNA also has a phosphodiester bond, right? We just looked at the structure. DNA and RNA differ only by one OH. Right, and there is also a small change in the base, which is in DNA there is called thymine, in RNA is called uracil. I didn't that is not very important for us, so I didn't talk about it. But so it is also doing this phosphodiester bond. Then there also, and in addition there are many other phosphodiester bonds, right? ATP, for example, has a lot of phosphodiester bonds. GTP has a phosphodiester bond. Now, enzyme, in addition to being a biocatalyst, that means in addition to increasing the rate of a reaction, it has to be very specific. That means if I give you 100 molecules, an enzyme should only act on the molecule that it should act on. Because in your body, in a cell, you have ATP, you have GTP, you have DNA, you have RNA, everything. But I want the enzyme to act only on the DNA, right? I don't want to disturb it because chemically they are all similar. They are all phosphodiester bonds, right? So that specificity has to come. So enzymes are very, or basically the major advantage with the enzymes is that they are highly specific and they increase the rate of the reaction many-fold. That means they are acting as a catalyst. That is something. Okay, I'll talk about the enzymes, but any enzyme that performs a hydrolysis reaction, that is called ACE, A-S-E. Okay, if you see any enzyme or if you see any molecule which ends with A-S-E, you see lipase, okay, lipase or nuclease. ACE means anything that performs a hydrolysis reaction. You know what are hydrolysis reactions, right? So you're basically adding a water molecule to that or then breaking into it, right? So any molecule that performs hydrolysis reaction is called ACE. Now, nuclease means a molecule or an enzyme that hydrolyzes nucleotides. These are called nucleotides. Okay, your DNA, right? Here DNA, these are called nucleotides. So any reaction, basically nucleases means something that hydrolyzes nucleotides. So these are called nucleases. So if you will say other kind of enzymes also, okay, which are ending with ACE, any molecule that ends with ACE is a hydrolysis based reaction. Now these nucleases can be of two types. They can be, as I told you, this DNA is very long, right? So the DNA is like, suppose it is a 3000 bases or 4000 bases. Now all of these are linked by your phosphodiester bond. So the nucleases could break it in the middle of it, okay, or towards the end. When you are breaking at the end, you shorten this by a specific one base at a time, right? When you basically do it at the end, those are called exonucleases. When you can do the hydrolysis reaction in the middle of the DNA, those are called as endonucleases. So there are exonucleases and there are endonucleases. So basically nucleases, so coming back to this, so I want to do a editing of my DNA, and DNA editing requires a cleavage or a breaking of your phosphodiester bond. A phosphodiester bond breaking is done by the hydrolysis of the phosphodiester bond, and any enzyme that performs hydrolysis reactions are called ACEs, right? Here, because they are doing the hydrolysis of nucleotides, these are called as nucleases. Okay, so this is the enzyme that we are mostly looking into, which are nucleases, and these nucleases could be either exo or endo. Now, as I told you, like enzymes are again like what do you think is the we said what are enzymes? Enzymes are basically they have to be highly specific. They are biocatalysts and other kind of the speed of the reaction. But chemically, what are enzymes? Everything is a chemical, right? So chemically, what are enzymes? Proteins. Okay, very good. And only proteins?
Sometimes nucleic acids.
Sometimes nucleic acids. Okay. So most of the enzymes are proteins. So there could be a small addition of either nucleic acids, or there is also one more very important biomolecule. What is that? Not lipid. Lipid is the important biomolecule, but enzymes do not have that. Carbohydrates. So enzymes are basically proteins, mostly proteins. Some of the enzymes have a small component of either nucleic acids or a small component of carbohydrates. So before we understand the chemistry of your phosphodiester bond, I want you to understand what are enzymes. Okay, and enzymes are basically your proteins. And I guess a couple of days back you had a lecture on protein folding.
Yes, sir.
Right? So you know how complex the structure of proteins are, right? You know how the amino acids fold into a beautiful structure, which is a quaternary structure, and give a nice architecture to that, right? So just again to revise that very quickly, so there are basically 20 amino acids, right? So the 20 amino acids form, like as I told you, you have a DNA, RNA, and then proteins. Proteins are basically your 20 amino acids in any random fashion. Random in the sense, every protein has a different sequence. They are all joined by a peptide bond. So, okay, all these 20 amino acids form these peptide bonds. So again, you see how beautiful it is in the sense, DNA is just 4 characters, right? And all the proteins which are few hundreds to thousands of amino acid long are only made up of 20. So there is not much diversity in the cell. Like in the sense that nature has to use only with limited resources, either 4 bases or 20 amino acids, but it has to perform some millions and millions of work, right? So every work requires a different protein, a different enzyme. So it actually just by using this 20 amino acids, it is modifying that, right? So now I guess because that lecture is already over, I will not go through that, but you have your amino acid, and 2 amino acids, when they come together, it forms a peptide bond, and when the multiple amino acids come together forming a peptide bond, that forms what is called as protein. So a protein with only amino acids, that is called as a primary chain. Okay, then because of the composition of the amino acids, it can take what are called as secondary structures and tertiary, it can take secondary structures and tertiary structures. Secondary structures are basically the folding of these amino acids so that they either look like alpha helices or they look like beta sheet. Now many of these secondary structures come together to form a much compact, because you can't have a linear chain. Then for every protein you will have like a 1 meter long chain. You don't want to have that. They form a very compact tiny structure which is called as tertiary structure. And many of the times, especially in enzyme, many of these tertiary structures come together and form what is called as quaternary structure. So basically you have proteins that come together and forms a functional. That means, for example, I'll talk about a protein called as, have you heard of something called ribosome?
Yes.
What is ribosome? Ribosome is something that synthesizes your proteins. But it is also a protein. It is like your chicken and egg problem, which came first, right? So ribosome synthesizes protein, but what synthesizes the ribosome? Now that is a different question, but ribosome is also protein, right? Now, but it is not a single protein. There are multiple proteins that come together and form a compact structure, and that is called as a ribosome, which is a quaternary structure. When amino acids fold, that is called secondary. When the secondary folds, that is called tertiary, and those tertiary structures are functional on their own. But if you want to do a larger chemical reaction, many of these tertiary structures come together and form a quaternary structure, which is called as which is quaternary structure. So all the enzymes that we look at have quaternary structures. That means many of these tertiary structures. Okay, so enzymes can perform various reactions. Like for example, this is a ATPase. Now, can somebody tell me what does ATPase do? As I said, ACE is something that hydrolyzes. ATPase is something that hydrolyzes ATP. So you have an adenosine triphosphate and it hydrolyzes the phosphodiester bond to basically this thing. And not only chemical reactions, many of the enzymes perform mechanical reactions also, which we'll not talk about. But just don't think that enzymes only do chemical reactions. They also can pull on things, right? They can also do mechanical forces and mechanical work also. Okay, so this is how an enzyme look like. Like somebody told, enzymes have a nice active site, right? So they fold in such a way that quaternary structure, like why is it that when you're doing a reaction outside it is slow, but when an enzyme does it, it's very fast? Why? Because it folds in such a way that it forms a nice active site where the pH or the ionic strength or anything is so conducive that it does a reaction, it increases it. You know what do catalysts do? Catalysts decrease the activation energy barrier. You are aware of activation energy barriers, right? When a reactant goes to product, it has to go through a activation energy barrier. So these catalysts make these nice active sites that basically the entire reaction, that the energy that is required to perform this goes down drastically. And so these are some of the examples. For example, these are all the amino acids. This is the structure of, you might think like, do we really need such a big protein or enzyme to perform reactions? Actually, the fold is important, but the reaction happens only in a very, very small region called active site. So this enzyme would have a small active site, and that is where a specific chemical reaction happens. Here is just, I'm giving an example of carbonic anhydrase. Can somebody tell me what does carbonic anhydrase do? Okay, yeah. So basically, what is anhydrate? Without water molecules, right? So basically, ACE, anhydrase, which also, that means it is doing a hydrolysis reaction to remove water from carbon dioxide, right? So that is how this reaction. So basically, this is I want to tell you, emphasize you, enzymes are, although they are big, they have a very small active portion which kind of perform all these reactions in a very specific way. So I just have some numbers here to tell you how efficient the reactions are. For example, any specific reaction you take. I'll take an example of a carbonic anhydrase or AMP nucleoside. You see, let me not go into the carbonic. You see this particular reaction, AMP nucleoside. If you leave AMP if you look at the reaction that it catalyzes, if there is no enzyme, the reaction would take around 69,000 years inside. If you don't have an enzyme, but if you have the enzyme, then it will just take 1 into 10 power minus 1. The rate is around 1 into 10 power minus 11 seconds. So you can calculate the, from the rate constant, you can calculate the rate. So yeah, so basically this is rate. So you see, for example, a reaction that takes 5 seconds is taking much smaller time. So enzymes enhance the rates drastically. You can say at least 1,000,000 times or more, so that everything is going as pre-planned. In the sense, you don't have reactions, you don't have to wait for a long reactions. And in addition to that, they also have a very high specificity. They'll only do the reaction in the area or the bond that you are allowing it to do. It doesn't do randomly or anything, and it also enhances. So you can see how enzymes are effective. Okay. Now, I think 4 o'clock is their coffee break, right? Yeah. So what I'll do is, I've started with genome editing, but I wanted to understand so that you all are on the same page to understand some basics. I've talked about DNA. I have talked about enzymes, because we are supposed to do a DNA editing, that is phosphodiester bond editing, using enzymes. So I hope you have brushed up your basics of both enzymes and DNA. Now when we come back, what we'll do is, how is this DNA editing actually done is what we are going to look into. So we'll assemble back at the normal time, 4:20, right? 4:20, 4:30 something, 4:20 I guess. So you can come back at that time, 4:20, 4:30. Okay.